Hi, my name is Shakked Halperin, and for my Principles of Synthetic Biology final project, I modeled a tunable and orthogonal oscillator. I hypothesized that a model of a CRISPR-Cas9 genetic circuit may reveal a method for creating a customizable oscillator. So synthetic genetic oscillators are useful for interrogating natural oscillating systems as well as building synthetic biological devices that may need to incorporate an oscillating system. And several genetic oscillators have been constructed, however, they require specific promoter-regulator pairings and are difficult to tune. The CRISPR-Cas9 bacterial immunity system might be used to create an orthogonal and tunable oscillator. The ability to implement this system to any set of promoters makes this oscillator highly customizable. And then, due to the ease of engineering guide RNA's binding location and binding strength, analysis of the system might reveal a highly tunable oscillatory behavior in terms of the amplitude and the period of oscillations. So CRISPR-Cas9 can be localized to a specific region of DNA as specified by the complementary 20 base pair recognition sequence on the guide RNA, or gRNA. While wild-type Cas9 catalyzes double-stranded cleavage of the DNA to which it binds, a mutant called dCas9 binds the sequence complementing the gRNA without catalyzing this double-stranded break. So dCas9 has been shown to be able to both activate and repress transcription when localized to promoters with gRNAs that contain a transcriptionally fused RNA hairpin that recruits a RNA binding peptide fused to an activating or repressing peptide. So the MS2 hairpin binds to the MCP peptide, and the COM hairpin binds to the COM peptide. The VP64 protein activates transcription, and the CRAB protein represses transcription. So oscillations can be achieved with an element A that activates itself as well as element B, while element B represses element A. Therefore, a way to construct an orthogonal and customizable oscillator might be to use a constitutive promoter, PA, to drive the transcription of an activating complex, which consists of gRNA1 bound to dCas9 and MCP VP64 fusion. That increases the rate of transcription of another constitutive promoter, PB. PA also transcribes an activating complex consisting of gRNA2 bound to dCas9 and the same MCP VP64 fusion. That increases the rate of transcription of its own promoter, PA. Finally, PB drives the expression of a PA repressing complex consisting of gRNA3 bound to dCas9 and ComCrab fusion protein. The dCas9 can be constitutively transcribed in a unit elsewhere. So the biosystem parts and compositors framework in MATLAB was used to investigate the production, binding, unbinding, and degradation of the system species with a temporal model. Since oscillatory behavior requires the activation and repression to act in a nonlinear fashion, the activation and repression must be the result of a multiple species cooperating dependently. While dCas9 bound to gRNA can be used to repress gene expression, the recently observed linearity of this type of system doesn't offer the necessary cooperativity for oscillations. However, the necessary cooperativity might be established if the dCas9 can be recruited to a site where it doesn't affect transcription until an effector binds to the gRNA. So it's been shown that dCas9 recruitment 200 base pairs upstream of the transcriptional start site shows little activation and repression, but significantly increases and decreases transcription when either VP64 or CRAB effectors were recruited by the gRNA. So assuming a quasi-steady state interaction, implementing this system should allow the activation and repression to be modeled by this Hill term. So the chemical reactions and the assumed Hill-like cooperativity of the gRNA dCas9 effector complex were used to create a system of 11 differential rate equations, grouped as rate equations 1 to 7. Due to initial lack of oscillations observed with the described model, with arbitrary variable initial values and constant values, I created a script that runs through a list of combinations of biologically feasible ranges of these constant and variable initial values and stored the combinations of values that yielded oscillations. I defined oscillations as three or more local maxima that have a minimum prominence of the potentially oscillating species mean value. And that successfully revealed combinations of values that gave rise to oscillations. So the first thing to note is that at the very least, gRNA1, gRNA2, and MCP VP64 mRNA had to be initially present, when only the repressive components, gRNA3 and ComCrab mRNA, were initially present. Immediate repression of promoter A caused the activating complex to never be transcribed, and therefore oscillations were not produced. Changing the initial value of, of gRNA1 from 0 to 10 changes both amplitude and period of the oscillations in the beginning of the simulation. However, eventually the parameters converge for all initial concentrations. So for the remainder of the analysis, initial values of all the species were set to 5 nanomolar. Now, changing the value of K-Cas between 0.5 to 2 nanomolar per minute does not change the oscillations, which was rather surprising. And changing the value of K-Crab from 0 to 3 revealed two interesting phenomena. One is that the oscillations are produced when K-Crab is greater than the value of KVP64. So in this case, KVP64 was set to 1. And that's why you only see oscillations when K-Crab is greater than 1. So when KVP64 was increased to 2, once again, only a K-Crab greater than the value of KVP64 yielded oscillations. And the second thing to note is that increasing the value of K-Crab decreases the amplitude and wavelength, while increasing the amount of time it takes for the system to reach a steady amplitude. So recruiting multiple ComCrab vectors with multiple gRNA com hairpins or changing gRNA target sites relative to the transcriptional start site might be the way to actually modulate the K-Crab to get the effects that you see here. Next, if we increase KVP64, you get an increase in both amplitude and period of oscillations, and decreases in the value around which, in this case, gRNA3 dCas9 oscillates. Modulation of KVP64 might be possible by recruiting multiple MCP-VP64 effectors with multiple gRNA MS2 hairpins, or by changing the guide RNA target site relative to the transcriptional start site. Increasing KdCas9 decreases amplitude and wavelength. KdCas9 might be easily tuned by changing the length of the guide RNA. So finally, we can look at all the species concentrations and see that the activating species, gRNA1, gRNA2, MCP-VP64, show an asymmetrical oscillation pattern. Their concentrations rise with the repressing species, gRNA3 and ComCrab, but quickly fall. This is because there is no transcriptional repression of the repressing species. Their fall in concentration is solely due to degradation. The transcription of the activating species, on the other hand, is quickly decreased as the repressive species concentration rises. So, in conclusion, this model of the CRISPR-Cas9 genetic circuit successfully produced oscillations when I got the correct values, and seems to have a tunable amplitude and period with parameters of the biological system that seem to be easily engineerable. So thanks for your time.